My name is Brenda Winkel, and I'm a professor of biological sciences at Virginia Tech.
And I'm Sherry Hildreth. I'm a postdoctoral researcher, and I work in Dr. Winkel's lab here at Virginia Tech.
And we're here to tell you a little bit about the research that we do in our group. We have been for many years interested in how cells work, and we are using plants to figure that out because plants are these wonderful experimental systems for studying genetics and biochemistry and molecular biology.
You're probably familiar with the term gene, and what we're interested in is how a particular gene corresponds to an mRNA and how that mRNA in turn corresponds to a protein. Proteins produce a group of chemicals called metabolites. So metabolites include pigments that you see in colors, and whether you have a red flower or a white flower, they produce the smells that plants give off, the fragrances.
And so what we've been doing in the lab is trying to figure out what all the parts are that are needed to make these very special chemicals, and then understand the rules inside the cell that govern how those processes take place. And so we're going to be telling you a little bit about some of the tools that we use in the lab, and then some of the tools that you guys will be using potentially to be doing experiments yourselves.
So we're very excited that you're going to be working with us and helping to identify different phenotypes in our AADL mutant plants. One of the ways that we detect difference in phenotypes here in the lab is by this piece of equipment, the HPLC. The HPLC is a great tool we have for both identifying and telling the differences in chemical compounds that the plants make. This is a great piece of equipment. It's used here at Virginia Tech and worldwide, and has led to many great discoveries. One of the most important things in science is not necessarily the individual tools you use, but how you use the tools you have. The power of observation is a great tool, and one that you're going to be using whenever you grow these plants and look for changes in the phenotypes in our AADL mutants.
So these characteristics of plants that make them so easy to study are their colors. The colors of flowers or leaves of seeds that make them very amenable to genetic analysis. And so if you take away one part of the plant or a gene that's required for making those pigments, there's very striking changes in how the plant looks. And that goes back to this power of observation. And so one of the things that we do is work with mutant lines that make altered colors or altered pigments. And the pigments that we're talking about are called flavonoids, which you may have heard of as well. They're found in all plants, and they're very important also for health and as well for protecting the plant from all kinds of things in the environment, including ultraviolet light and pathogens and predators, but also for attracting birds and pollinators and other organisms that are beneficial.
So we have a batch of seeds that we are growing right now, and they are mutant lines of this brand new gene that we're working with. See, the plants are growing and the seeds are setting right now, and we'll be shipping them to you shortly. What we're asking you to do is for you to grow these plants and notice and observe what happens to them over time. The plants could have things like a change in color, like these plants here beside me. As they've gotten older, they have become stressed and their color has changed from green to purple. As you grow the plants, notice if there are changes in the size, in the color, and how fast they grow or how slow they grow. Anything that you notice is important to us and will help us in our research. So we look forward to working with you and hope that this is as fun for you as it's going to be for us.